We've been using MRI machines for the last 10 to 20 years to try to look at brain function and starting to look at brain connectivity. What I do is look at how the different parts of the brain work together to produce behavior. And I think that's the most simple way to think about the connectome. It's about how does the brain work together.
In our Charlie Rose Brain series, we have explored our magnificent human brain through two remarkable series. With my co-host, Nobel laureate and Columbia University professor Dr. Eric Kandel, we have brought together many of the world's best scientists and researchers on brain science. We continue that mandate looking at a new project by researchers at Washington University in St. Louis. James Gorman writes in today's New York Times about the team's work to develop the first interactive wiring diagram of the living human brain. This data could help us answer this important question: how do differences in brain wiring relate to differences in our behaviors, our thoughts, our emotions, and our experiences? The research is part of the Human Connectome Project, a roughly $40 million five-year effort supported by the National Institutes of Health. James Gorman joins me now, and of course, with me on this journey to help understand this research and its potential impact is Eric Kandel, Dr. Eric Kandel. So I am pleased. I mean, I with great excitement, James, I read this morning and then called him up. It's like we're back in the saddle again. Right?
Brain science is alive.
You know, so, I mean, it was just so I mean, you gave us a great moment.
That's great.
Tell me about what led you to this, because not only, you know, did you write, but you were a guinea pig.
Yeah. Well, we've been thinking that, I mean, we cover, you know, advances, news stories in neuroscience often, but we had been thinking, as many people have, as you have, that we ought to do it in a little more systematic way. And so a while ago, my editor asked me to start looking not so much into treatment and some of the sort of applied research, but into the nitty-gritty, you know, how neurons connect, what's going on in basic brain science. It's obvious that that's a huge, the technology is changing, some money is flowing into it, there's a lot of excitement, and around the same time that we were getting going, President Obama announced an initiative, a grand challenge. And so we thought, well, how are the ways we're going to look at this? And one of the big challenges is that trying to cope with neuroscience is like, I mean,
tell me about it.
It feels bigger than the universe, you know.
Yeah.
It's just so, all we could do was do a sampling of some of the projects. And we thought, well, let's start out with mapping. It's not everything, but it serves as kind of a foundation. Let's talk to some of the people who are trying to map the brain in different ways. And the question was whether we were going to start sort of at the level of the electron microscope, very small, or at the level of the whole human brain. And the whole human brain is something that people relate to immediately because they have one. So that's where we started.
We know about the mapping, Eric, of the human genome, you know, and now we talk about the mapping. Give me your sense as a neuroscientist of the significance of the things that he reported on.
I think the significance of this is severalfold. One is we want to understand how the human brain works, and a key to understanding them is how the different components interact. Many people have been looking at this from a variety of perspectives, doing imaging studies. This particular study moves it forward in the following way. One is it is very systematic. It doesn't take the three of us, it takes 1,200 odd people, number one. Number two, these are people from normal walks of life, men, women, baseball players, academics, etc.
Who have their brain scanned.
Who have their brain scanned. And Dina is going about this in a very systematic way. She wants, as Jim pointed out in his article today, to work out in general the connectomics of the brain. And she's doing this by doing four kinds of studies. One is she just wants to do functional imaging to see, you know, what areas of the brain are active. Two, she wants to see how areas interact, and she's got two ways of approaching it. One in the resting state to see what areas are active together, and then she has a way of actually tracing connections, connectomics, between them, what she calls the pathways, the white matter in the brain. And finally, and critically important, she's going to combine these three imaging approaches with behavioral studies. She's going to see what happens when you learn something, when you sing a song, when people interact socially. So she's going to get a variety of different perspectives on this. So what is unique about this is not that any component of it is new, but it's systematic. And that's one of the purposes of the brain project.
And at the end of this, about a year from now, it'll be available online, and people can...
Yes, it's already, much of it is available online. They release a certain amount each time, and they have a web-based program called Workbench. Any scientist in the world can go in there and essentially query the database and look for connections between different activities in the brain or different qualities that the people may have.
I think there's another very nice thing about this, and that is it's a wonderful environment in which she has done her work. Marcus Rakel is there as one of the pioneers in brain imaging. David Holtzman, an outstanding neurologist who we had here in the series. David Van Essen. It's a wonderful environment in which to work, lots of interesting people to talk with and learn from. So this is going to be a thought-through and reliable set of studies.
And as she would be the first to point out, she's one person working on one part of this.
Yes, she actually comes from psychology.
Yes, right. That's exactly right. She comes from psychology.
So many people involved.
So interesting, the psychology, because, I mean, there is this sort of series of questions, too. You went through this.
Yes.
Tell me what it was like.
Well, the MRI, as anyone knows who's gone through one, is very noisy and confining, and most people have, in addition, to cope with the fear because they're usually there for some sort of diagnosis. So that was one thing. The tests that you do in the MRI, there's a variety of them. Some are as simple as curling your toes to map where your toes are. Others require a kind of memory activity that made me convinced that I had completely lost my mind.
Really?
I suppose it's
asking about what you remembered about...
Well, you see a series of pictures, it's called, and you try to remember whether the one that you saw 2 times ago or 1 time or however, this was 2 times ago, is the same as the one that you're seeing now. So you see a pipe, you say, okay, I saw a pipe, then a tree, okay, a tree, a car, and then you have to push. Is that the same as the one? No, it's not the same. And then a pipe, well, yes. No, that's not. And pretty soon it's very hard to do. And one of the fascinating things that she said to me afterwards was, I said, I was working so hard on that. And she said, well, that's one of the things that this might show, because you're going to have your emotions are tied up in competing in this test. That's not going to be true for everyone. Some people are going to be very different. They're going to take it much more easily.
You write, neuroscience does not have a baseline database for structure and activity in a healthy brain that can be cross-referenced with personality traits, cognitive skills, and genetics.
That's exactly right. We don't have that.
And how important is it to have that?
It would be marvelous to have, but this is not around the corner.
It's within the next 10 years?
We talked about this once before. My feeling is, to a complete understanding of the human brain, we're speaking more like many decades. Helen Mayberg, whom we had here.
Oh, we love Helen Mayberg.
And you mentioned very prominently, who commented on this thing, has done wonderful imaging on depressed people and delineated the beginnings of a neural circuit of that. If you look at psychiatry, for example, there is no area, no disorder that is really understood on this level. In fact, in schizophrenia, autism, we're just beginning to understand which areas are critically involved.
We have not done this, have not been able to do this before because we did not have the tools to do it like we do now? The imaging tools? Or is it some new theory of...
No, there is no new theory. The imaging tools, when they came along, were noisy and not completely reliable. And it took a long time to really work out the difficulties with them. They're now much more reliable, and this is a very reliable group of investigators. That's number one. Number two, you need resources. You need huge resources to carry out these large-scale studies. And three, the problem's extremely difficult. She's not asking herself, what is the cause of schizophrenia, which would be a very difficult problem to tackle right on. She's asking herself, how does the brain function in very general terms? How do people handle the same task in somewhat different ways?
Explain this to me, because you said in your piece, the central question is, how do differences between you and me and how our brains are wired up relate to differences in our behavior, our feelings, and our emotions? I've said this in the introduction.
Well, one of the things that I did, for instance, which all the volunteers go through, is you fill out a questionnaire,
Right.
and interestingly, the person, the research assistant who's there, leaves the room because it's private. So how often do you feel sad? Do you feel discouraged? So it's trying to get at it, and these are very traditional, well-used psychological tests that many people have used over the years, so they can sort of tie them to what that says about your state of mind. So if you have 1,200 people and you have data on how they take these tests, you may be able to see what the variations are that correspond to the variations that show up on the test. Maybe not, but you may be able to see something closer than what you have before, and that may begin to give you an idea. The other thing is that you're not just looking at a difference in size or structure, you are looking at what happens when people are doing something, which parts of the brain are
Patterns
working
of
together.
interconnectivity.
Yeah.
Patterns.
Explain
I give you a simple example. This would not apply to New York cab drivers
Right.
who get lost all the time. But in London, you have to pass an examination.
Well, you've actually said this before, but go ahead.
And if you look at the brains
Yeah.
of London cab drivers, their hippocampus, the part of the brain concerned with memory for space, gets larger the more they drive.
Yeah.
And when they stop driving, it shrinks a bit.
Yeah.
So this is wonderful, but we don't know what are the other areas that this hippocampal region interconnects with, and these are the kinds of things we can find out. I mean, we know in individual cases, if you just oppose your fingers many, many times, or if you play the violin, your representation of the left hand, which does the fingering, is much larger than for people who don't play a violin in the motor area. So we know in very primitive ways that different body functions are represented in the brain, and they change with experience. But a systematic
And
study...
use.
And use, yes. And they shrink. And of course, we know with certain disease states that certain areas are involved and not others. But a systematic study of normal behavioral patterns, this is a wonderful beginning. This is not the end. I think Barch would be the first one to tell you this is a systematic beginning.
Yeah, but I mean, it's a heck of a beginning.
It's a very serious
and it's a lot of great universities. I mean, there's one program that's got, a couple
the
universities.
Mass General and Harvard are developing imaging techniques.
With UCLA, I think they're involved with UCLA. Then
Yes.
Oxford's involved with Minnesota and somebody
Yes,
else.
yes.
And
Yeah.
Washington University.
And Washington University.
Yeah. It's a very serious
So
program.
across, and you just got $200-300 million to do what?
Mort Zuckerman gave Columbia $200 million for Mind-Brain Behavior Initiative.
So what's that?
We want to tackle problems of the brain on a variety of different levels. We want to understand how the brain works in simple animals as well as complex. We want to understand disease states. We want to understand how mental functions have an impact on other aspects of society. For example, how do people respond to work of art? What is it about music that really moves you in such a way? So to use the Mind-Brain Behavior Initiative to interact with other components of the university.
Sort of understanding what makes us human, isn't it?
That's right.
Yeah.
That's right.
They're also involved in, I mean, I assume part of this is not the mapping thing, but maybe it is also, it's the connection between neurons, right?
Yes.
Yeah.
They're not looking at this level with the MRI at the connections
Right.
between neurons. They're looking at the connections between regions
yeah.
and information flow and the direction of information flow in the brain. There are other people that are looking at the connections between neurons more in mice and also in flies.
Fruit fly.
In fruit fly,
Yeah.
Jerry Rubin is doing a parallel thing at Genelia Farms at the Howard Hughes Medical Institute, in which he's systematically mapping the brain of the fruit fly using genetic and anatomical techniques that are also quite spectacular, and will be applicable to higher animals as well, and is combining this with behavior. So what we talked about the last time,
Right.
which is
The
brain
President's initiative and other things, right.
This is what you're seeing in action. People are doing things that they did before more systematically, more collaboratively, in order to push this enormous problem forward.
And are they getting more money?
No,
Really?
no. The money is embarrassing.
You mean in terms of, you're talking about money from NIH, or are you talking about money from...
$100 million a year at the moment is set aside for the Brain Initiative. That's not
$100 million
enough
a year?
to pay for dinners for the three of
And
us.
it's not, it doesn't seem to be new money. Oh, I could get by.
that-
Yeah, we'll get by on $100 million. Yeah, but I mean, the point's well taken, though. $100 million seems like when you think about...
Well, NIH, its budget for neuroscience, apart from that, is $5.5 billion.
And
Neuroscience.
It's part of this is coming out of pre-existing funding.
Yeah. And what makes you crazy when you think about that is because it is about understanding a whole range of disease
It's an enormous problem.
that affects so many millions of people's lives, whether it's depression or...
What Francis Collins would tell you, the head of the NIH, who's pushing this very hard, is that there was a small amount of money at the very beginning of the Human Genome Project, and it served as kind of a spark to develop some techniques and seed money. So that's the hope. You know, we'll see. In Europe, they've put $1 billion out for a project that is, at least the people I've talked to, not as well regarded as the plan that NIH has come up with.
It's not as focused as empirical. It's really trying to use...
What's it focused on then?
It's trying to see whether you can simulate the human brain in a computer.
What do you think of that?
And since we know so little about how the brain works, it's going to be hard to simulate it. But, you know, there are other people joining in, and it's going to interact.
But is there a complete sharing of information across the board?
Yes.
What you're doing and what Helen is doing, what everybody else is doing?
That is not a problem.
That's really...
The problem at the moment, in addition to human intelligence and energy, is resources. The resources are grossly inadequate for the job. I think everyone would admit that who's involved in it.
And what's interesting about this is three-dimensional, too, and interactive.
Yes.
Oh, yeah. It's,
explain why that's important.
Well, it makes it so complicated that you need new kinds of mathematical tools and software, essentially algorithms, to understand. At Harvard, they're working on the Connectome at the neuron-to-neuron level. And you can, the Jeff Lichtman, who works there, is fond of saying, well, you know, the smallest volume that we can see with the electron microscope is in the nanometer range, and the volume that you see in the functional MRI, which is what they're doing, one is a trillion times bigger than the other. And the one that's the trillion times bigger is one... I may get my numbers wrong here, but it's something like 1/1000th of a cubic inch. It's a very small. And then you've got the whole brain. And at every level in between, and correct me, I mean, I'm just talking about at every level in between, there are important interactions going on at that level and between levels.
Absolutely.
So
this is why it's very important, and this is happening, to do the human brain in parallel with the worm, with the fly, and with the mouse, because they can give you solutions to the synaptic logic.
System is so simple.
Much simpler, but they have, in many cases, very parallel behaviors.
They're also, you can explore them in ways that you can't explore the human brain. So you can take a fly.
We should also point out that this is not the only methodology that's come along recently. We have optogenetics.
What's that?
Optogenetics is a way of inserting a gene into a cell so that it will now respond to spots of light. And you can use that to turn on any cell combination of cells that you want, and you can shut cells off by putting in a gene that shuts off the cell. So this is a very powerful technique. You can use it in intact behaving animals, and it's really revolutionized the field.
Has all of this attention, perhaps not money, but attention, from my little television series with you to New York Times doing all this stuff, has it spurred enormous interest in young physicians, both clinical and researchers, to become neuroscientists?
You cannot imagine. My wife, Denise, showed me recently a graph of the evolution of graduate education in the United States. For all disciplines, it's like this. For neuroscience, it's like that. It just keeps on increasing. The problem is that with the cutbacks of funding for basic research, this is discouraging people from going into science. And young people are having more difficult time, talented young people having more difficult time getting a job than it did before. When I came along, if you could read and write, you got a grant. Now it's a major accomplishment.
Well, you could read and write then, couldn't you?
Barely.
One of the problems with grants is that the political need is to say we're going to cure a disease that hurts you, and the level of the science is to say, well, I'd like to know how this neuron in a fly detects motion from left to right.
Terrific.
Yeah, exactly.
I'm quoting Sebastian Sunk, so who I'm
Sebastian. Yeah.
He's a Connectome person.
Yes.
Yeah. This is very exciting stuff, and we're learning things. And there's a lot going on in a variety of different places. And yet some of it has enormous connections to it. It's about some basic questions that are referred to.
And it's going to bring in other sciences.
Yeah.
As you mentioned, engineering, mathematicians, chemists are going to come into the field, nanotechnologists, because their help is needed, and they find these problems interesting. Not to speak of genomicists.
Yeah. But it also reminds me, little old me, of how, as much as we're learning, the vastness of what we don't know. And the extraordinary quality of this brain is really amazing.
Yes.
And you have to always ask yourself, don't get ahead of yourself, in a sense, as you gaze at the future, because this is hard work. This is
Absolutely.
It is step by step by step.
But it's very satisfying.
Yeah.
I mean, after all, you're tackling the most fascinating problem in all of science.
Well done, James. Well done. Thank you, my friend.
Thank you.
Great to have you here.
Pleasure to be here.
Back in a moment. Stay with us.